All right. Well, I'd like to welcome you all to the virtual open house at Lehigh University. My name is David Zappulla. I'm going to tell you a bit about the Zappulla Lab research program focused on telomerase, telomeres, and senescence. So, first I need to tell you a little bit about the end replication problem. Why should you care about this and what is it? So, the end replication problem refers to the replication of the ends of eukaryotic linear chromosomes. So when DNA synthesis occurs in S phase, there's a leading strand and lagging strand, as you all know from your science courses. And the leading strand proceeds to the end of the chromosome, but the lagging strand is discontinuous replication. And so when these RNA primers shown in blue are removed, ultimately, you're still left with a gap in replication at the termini of chromosomes. And this incomplete replication adds up and causes problems. So in humans, the enzyme telomerase, which maintains telomeres and overcomes the end replication problem, is downregulated early in development. And so throughout your lifespan, telomeres are shortening. And this shortening of telomeres throughout the tissues of your body is also concomitant with an increase over lifespan in disease incidence. And many different diseases you can think of that become a greater concern as you get older. One of those is cancer, most famously. And so you can envisage telomere shortening as being sort of a tumor suppressive mechanism because it overcomes the ability of cells to grow out of control. And therefore, in order to grow out of control, cancer cells have to overcome the end replication problem. They do so in 90% of cases by upregulating telomerase, and in 10% of the cases, they use an alternative or alt mechanism for maintaining chromosome and telomeres using recombination. Okay, so I and my lab members are fascinated by this relationship, sort of yin-yang relationship between aging and cancer, and how telomeres and telomerase are at that interface between the two, with telomere lengthening promoting longevity, but also being proto-oncogenic. Okay, so I've mentioned the enzyme telomerase, this Nobel Prize winning enzyme. What does it do? What is it composed of? Fundamentally, it's composed of a catalytic protein subunit called TERT. It's a reverse transcriptase, which means it reads RNA into DNA, and it also has an RNA subunit, so it's sort of like a reverse transcriptase with its own genome, essentially running around adding DNA at the ends of chromosomes, using its templating sequence of RNA to allow the synthesis of DNA at the 3' end of a chromosome. The enzyme then translocates and does this process iteratively, adding repeats of DNA based on the template, and then it's also coupled to recruitment of DNA polymerase alpha and delta, and synthesis of DNA on the other strand. So it's sort of a one-stop shop for chromosome extension. So it's that enzyme that overcomes the end replication problem. And three of the major questions in the telomere field that my lab is addressing are as follows. What is the physical organization of telomerase RNA, that squiggly piece of green that I just showed you in the prior slide? And how does it relate to function? That's something that I've been looking at myself and with my own lab for nearly 20 years now. And then how is telomerase regulated to maintain telomere length? And finally, what are the cellular consequences of telomere loss? So my lab's research program basically boils down into three parts that address those questions. Again, I mentioned telomerase RNA protein complex enzyme organization, schematized here. The way that the enzyme is maintained to take telomerase to the short telomeres that need extension, and what happens without telomeres and, for example, due to loss of telomerase, telomere shortening, and that phased process as cells go into senescence and then can escape it either by telomerase reactivation or by recombination-based telomere maintenance. So I'll tell you a little bit about each, and then I've got to wrap it up. So The organization of telomerase RNP, one thing that I've been studying and my lab has been studying for some time now, is the hypothesis, now sort of a more established model, that telomerase RNA, at least in the budding yeast Saccharomyces cerevisiae, which we study predominantly, is a flexible scaffold for holoenzyme subunits, which are the proteins that bind and protein complexes that bind to the towards the more distal parts of these three long RNA arms that radiate away from the catalytic core. This is the secondary structure that I was able to solve as a postdoc in Tom Cech's lab years ago. And we've shown that all of the holoenzyme-specific subunits, other than the blue TERT catalytic subunit S2 in yeast, can be repositioned to novel places in the RNA, shown by all these different arrows, and still maintain their function in the cell, which is just striking and very novel. And so we've been sort of exploring this idea further and further, that telomerase RNA is a flexible scaffold for subunits, a very different paradigm for RNA protein complex organization, much different than the ribosome, for example, which you may know something about in structural detail, much more ordered in three-dimensional space. We've also, over the years, discovered three different essential structured, well, at least largely structured domains within telomerase RNA in yeast, and as well as some in humans. And these domains are very interesting to us as well, and we're exploring them further to see what they contribute to mechanism, which we've gotten very interested in telomerase. Okay, so to introduce the second part of my research program with one slide, one more slide, I'll just say that, in eukaryotic organisms, which constitutively have ends, we need telomeres to cap them. They're basically DNA repeats, as I mentioned, but they're also bound by proteins that bind to the telomeric repeat sequences specifically. And these telomeres need to be maintained at a certain length at the end of every chromosome to do their jobs and cap the natural ends of chromosomes. So to do that, telomerase, shown in the left here, needs to be preferentially recruited to short telomeres that need extension. Because telomerase doesn't act in every cell cycle on every end, it's recruited to short telomeres, extends them substantially, and then they whittle away due to the end replication problem over many cell divisions before telomerase visits that end of that chromosome again. So we're interested in the mechanism by which this occurs, and we made a big breakthrough in this regard about 5 years ago in my lab, where we showed that the Ku subunit of telomerase in yeast binds to the protein SIRF4 in order to recruit telomerase to seemingly to short telomeres preferentially. And we're working on that, the details of that now. And it's a really compelling molecular mechanistic model because it shows how this pathway fights with RAP1 interacting factors 1 and 2, which have been long known to dominate long telomeres. And so we think that the explanation is that it is competing with the recruitment pathway that wins out at short telomeres. But it keeps, the RIF proteins keep this recruitment from happening at long ends. So we're fascinated by this. We're exploring it extensively with an NIH R01 grant funding that we have, and really excited about where this is headed to. Okay, and then to introduce the third arm of our research program, I will just mention that, unlike germline stem cells in humans, talking about humans now, if you consider lifespan and aging, telomeres shorten in the rest of the body in tissues, and throughout that process, these stop signs indicate senescent cells that have gone into cell cycle arrest due to very short telomeres. And so we're interested in this process, and we were using yeast as a model system to explore this general system. And so in yeast, the same thing that I just showed you occurs if you delete a telomerase subunit. And we've done RNA-seq studies in duplicate compared to wild-type controls at 5 different time points through senescence and then into survivorship to explore the transcriptomic response to telomere shortening, which approximates aging, at least in humans, and then this other process, which is survivorship, as I mentioned. So What we find from this preliminarily is that nearly half of the genome is transcriptionally altered, either down or up in the process. We find phases. Each of those 5 points that I described represent a different stage of senescence or survivorship with a distinct transcriptional response. So we're interested in understanding the regulation of this process and what the different subgroups of genes are doing, enriched pathways of various types, including autophagy, for example. And then we're also looking at, which is really in our wheelhouse, looking at the hundreds of new link RNAs that we've discovered that are differentially upregulated in senescence. And that's been a really exciting thing for us to evaluate, at least bioinformatically, and now we're moving into wet bench experiments to get into the role of these and genes involved that are protein coding in the senescence checkpoint itself and the survival of cells through the senescence process and so forth. Okay, with that, I'll just wrap this up and just mention my email and my phone number. Please get in touch if you're interested in talking about any of my research further, or about Lehigh in general, or anything else. We're excited to have you interested in our program, and would be happy to Zoom ad hoc if you're interested in doing some of that in the coming weeks as you come up to the application deadline. And reach out, get in touch, and very excited to be here at Lehigh, and hope you might decide to join us here as well. Thank you.